35. Don't freak out about genetic engineering. We've been doing it for a long time. And then these cells, these two kind of just, I don't know, gratuitous slides about cells, fact sorting of cells, increasingly important in developmental biology, using fluorescent antibody tags. Not easy to grow these cells, got to have special media supports. Okay, how are DNA molecules analyzed? Okay, restriction into nucleases. See, Madison? So, bingo, yeah. I don't think I ever actually said it today. And so they cut internally at specific sites. You can get these discrete fragments. You can analyze them by electrophoresis, gel electrophoresis. You can analyze that by radiography. You can sequence big pieces of DNA. How's it done? Fred Sanger's dideoxy method. So this was very clever. He wasn't taking something and chopping it up the way you do, like the Edmond degradation of a protein from the N-terminus. You don't take an intact piece of DNA and chop it up. You make something. You make a whole population, a family of truncated products, and then you fractionate those on a gel. So you use these dideoxys because you need the 3' hydroxyl to attack the alpha phosphate of the incoming nucleoside triphosphate, right? So if you don't have that attacking nucleophile, it terminates. So you sprinkle in a little bit of the dideoxy for each of the 4, you get truncated products. These things are single-stranded. You run them out on denaturing gels with urea because you don't want to you don't want to look at the heteroduplex. You want to just look at the truncated form that differs by one nucleotide, one base from the shorter one. So you just look at these ladders that are separated by one base. So they're truncated, single-stranded products, and then we used to analyze them by running these 4 lanes. Now you use these fluorescent labeled dideoxys, and you can actually do all of this in one tube, run it on one lane, and then scan it in an automated fashion. I mean, this is for the genome project. So you need, for that, and you get these chromatograms. Okay, hybridization, without it, most of these techniques wouldn't be possible. Southern blotting, we've seen that often. You blot, you probe with your labeled probe. That's the old school way. Hardly anybody does that anymore. Now what's done are these microarrays with synthetic oligonucleotides on a chip, and then you use different probes, and then you can readily ask for hundreds of genes what's going on with that cell type. Okay, so those microarrays. In situ hybridization, 2 types, on chromosomes or in cells, right? So for chromosomes, then you can find out where is that piece of DNA on the whole chromosome. You use these fluorescent tags. For cells, you want to know which cells are expressing an RNA in a cell layer or in a tissue section or at a certain time. And so you do this by in situ, in place. Cloning, okay? We don't want to just chop these up and run them out on a gel, but we want to make a whole lot of just one piece. So we use the site-specific endonucleases to make these sticky ends and that we can recombine with DNA ligase. Then we put these in plasmids that have an origin or replication, a means of selection. Then these can be grown up in bacteria. And then we can expand this then to make libraries, genomic libraries or cDNA libraries. We can go back and fish it out using a type of hybridization. The cDNA libraries, to make those, we have to first use reverse transcriptase, right? But that's not enough. We need to go back because we only have a single strand of DNA then. So to clone it, we have to go back with DNA polymerase to make it double-stranded. Now we can clone it. And then we can make libraries then of genomic DNA or cDNA And the advantage of that is then we can find the introns because we can compare the two. Then polymerase chain reaction, pretty straightforward how this works. You melt, you anneal specific primers that you were able to synthesize because you knew the target sequences. And then you just go through these rounds of amplification, this logarithmic amplification, and then you end up with primarily one product. So you start out with so little DNA you can't even see it on a gel. And then you do this like 25 or 30 times, and then you can run a gel and you actually see one band. Unless it was a crappy reaction and your primers weren't specific or it wasn't, you know, and you just get a smear and all this stuff. So that kind of takes a while to sort out. Okay, what can you do with it then? So you can use PCR then to make these libraries, right? Genomic or cDNA libraries. You can use it then to test for HIV, viruses in blood. You can also use it in forensics to generate a molecular fingerprint then